Hello, everyone. My name is Joe Barco, and I'm Senior Director of Workflow Marketing at Unchained Labs. I'll be your moderator, and thank you for joining us today. We'll have a Q&A session at the end of the presentation. Please type your questions, and we'll get to as many of them as we can. And now I'd like to introduce Kevin Lance, the Marketing Manager for the Analytical Portfolio at Unchained Labs. Today, Kevin will introduce us to a powerful combo of instruments for gene therapy development. Big Tuna, Stunner, and Uncle together can help you efficiently characterize AAV constructs and formulations. And now I'll hand it over to Kevin.
All right, thanks for that introduction, Joe.
Sure.
And thanks everyone for joining me here today. So I'm here to talk about how Unchained Labs Gene Therapy Squad can get you hassle-free AAV and LNP sample prep and characterization. Getting almost anything done with AAVs and LNPs takes too much sample and time. That's why we developed the Gene Therapy Squad. The squad is a set of tools designed to deliver unique insights and faster results on AAV and LNP samples, helping you to solve your next problem faster. We'll check out some examples for AAV first and RNA LNPs a bit later. Here's the Gene Therapy Squad: Stunner, Uncle, and Big Tuna. Let's start with Stunner. Stunner delivers easy and efficient answers for AAV with capsid titer, empty-full ratio, and aggregation results using only 2 microliters of sample. Let's dig in to learn more. When it comes to Stunner, we love to say that it helps you know your AAV inside out, because Stunner is willing to do whatever it takes to get you the full story about what's inside your AAV. And characterization of AAVs is a very broad subject, and it's still evolving. And for every one of these needs, there are many technologies trying to meet that need. Today, I'll show you how Stunner can help address the needs shown in green. That's titers for full capsids and total capsids, empty-full ratio, and if there's any aggregation going on in an AAV sample. Let's focus in on 2 technologies first, DLS and UV-Vis. Each of them tells a part of the story. DLS gets you info on capsid titer and aggregation, but can't say anything about what's happening with DNA. On the other hand, UV-Vis gets you the total amounts of protein and DNA and can link those to empty-full ratio, but doesn't tell you any info about if this is contained inside of capsids. So each gets you valuable info, but neither tells you everything. What if you could combine them to get the whole story? That's where Stunner brings it all together to deliver answers on capsid titer, empty-full ratio, and aggregation with only 2 microliters of sample in less than a minute. And there's no labels, reagents, or standards. You just use the sample you've got. So how does Stunner do this? Simply add in 2 microliters of AAV above a titer of 10 to the 12th into the input well, and it's pulled into the microfluidic reservoir. At this step, the sample can sit for up to 2 hours before reading with no concerns about evaporation. After the plate is loaded into the instrument, the circuit is read first when empty. That removes any absorbance contribution from the plastic. Then a pump applies a small vacuum which moves the sample into the microcuvettes to be read. Because the circuit has fixed path lengths, it can make very precise and accurate reads. Both DLS and UV-Vis are read on the same sample in the larger sample microcuvette. Here we can see exactly what sample loading looks like with the help of some fluorescein and a black light. In 2 wells, we've already added 2 microliters of fluorescein. As you can see, to load another sample, you simply add in 2 microliters into that input well, and the sample wicks into the plate. Checking out your AAV with DLS tells you what shape your sample is in, if there's only capsids or if aggregates have started to ruin things. DLS also lets you figure out how much SLS intensity is coming from just your AAV capsids. That's shown as just the green peak on the left of the slide, while the gray peak is aggregates and larger stuff. UV-Vis brings information on how much total protein is in your sample and how much DNA is in there. Based on some simple information you tell Stunner before an experiment, it then does all the heavy calculating for you to figure out empty-full ratio and percent full. What Stunner is doing is shown step by step here. First, DLS identifies the capsids, the aggregates, and how intense the signal is from each of these populations. So that might be the area under the curve for the green capsid peak versus the total area under the curve for that DLS result. In the middle, that info then helps you split up your SLS signal from a total value into the amount that's just attributable to that signal from empty and full capsids combined. And finally, UV-Vis brings in that info about empty-full ratio plus protein and DNA totals to provide the last piece of the puzzle. Here's the whole story told in one graph by Stunner. Starting with the left side, the blue bar, which is the protein in a sample. So UVViz got you the total amount of protein, and here we're expressing that as how many possible capsids all that protein could make. By adding in DLS and SLS info, Stunner then splits up that information into the light blue bar, which is aggregates and extra protein junk, and the dark blue bar, which is the total titer for the AAV capsids. On the right-hand side, in the green bar, the process follows the same steps for DNA. The total height of the green bar is the total amount of DNA expressed as the total number of viral genomes that you can make. Light green is extra DNA, and dark green is the number of full AAV capsids. Dividing dark green, which is full capsids, and dark blue, which is total capsids, gets you to a percent full number that we also convert into an empty full ratio for you. Here's that same data showing how Stunner helps you know your AAV inside out. Dark green divided by dark blue is percent full, which is shown in yellow, and purple is percent empty, which is just the remainder. An empty full ratio is the ratio of those two values. You can see in the tables below the bar charts where we've quantified the data shown by these bars. For getting capsid titer, Stunner is way faster and requires less upfront work than ELISA's. Here we've taken full or empty AAV9 and ran a dilution series where we've compared Stunner's total capsid titer metric against the results for a capsid ELISA. The results have slopes close to 1 and R-squared values above 0.99. And importantly, it would take hours to get this ELISA data, while Stunner's done it in less than an hour with no sample prep and tons of walkaway time. In collaboration with Stride Bio, we've compared Stunner versus ddPCR results for genome titer for two AAV types, single-stranded AAV on the left and self-complementary AAV on the right. The genome titer of the single-stranded AAV sample was characterized using a robust variety of ddPCR experiments to best capture the average titer, while the titer error bars on Stunner come from a single run of triplicate samples. For the self-complementary AAV sample on the right, the optimized ddPCR protocol was used and both compare very favorably to quantification by Stunner. To look more into our ability to see percent full values, we took stocks of empty and full AAV9 and mixed them at different ratios. For each combination, 100% full stock, 80% full stock, etc., Stunner gets a good read on the data, and percent full and percent empty align well with the expected values based on how much volume you've pipetted. Working with Stride Bio again, we examined how Stunner's quantification of percent full compares with AUC for a highly purified sample. AUC was read for the highest enriched percent full sample, and then dilutions with empty capsids were used to create the target percent full. AUC and Stunner were then measured, and they correlate very well, and both are highly linear, as shown by their high R-squared values. Importantly, AUC requires orders of magnitude more sample than Stunner. It takes way longer than the 45 seconds a single Stunner read needs. And this idea actually works for any serotype really easily. Just tell Stunner the basics about what the serotype you have and what is its DNA payload, and Stunner takes care of the rest of the heavy calculating. So from only 2 microliters, Stunner combines UV-Vis, DLS, and SLS data to tell the whole story about your AAV, delivering mountains of data about sample aggregation, AAV full and empty capsid titer, and empty full ratio. So that's how Stunner delivers label-free, standard-free, and hassle-free gene therapy quant. Now let's check out UNCLE, which helps solve AAV stability problems. If we think of AAV as a piñata, the DNA is candy, the capsid is cardboard and colored paper, then this is what a thermal stability experiment looks like. UNCLE is the broadsword-wielding warrior who's wasting no time seeing what this capsid can withstand. Meanwhile, gene therapy researchers are there in the background, cheering on the experiment and excited for the results. So UNCLE uses the results from a thermal stability experiment to look closer at 2 questions that are really important to AAV development. First, is my virus aggregated? And second, is my virus capsid going to be intact? Knowing the answers to these questions is key to optimizing capsid design, formulation, and process and storage conditions, since you want to deliver an answer that keeps your virus in the best shape possible for the longest time possible. You can also ask if stability problems are the underlying cause for some potency, purity, or immunogenicity issues. How Uncle does this is by first being an all-in-one stability platform built for biologics and AAV that combines 3 different detection technologies to look at your sample. It uses 2 lasers to excite either label-free protein intrinsic fluorescence or a variety of dye-based fluorescences. With full spectrum fluorescence detection, Uncle can pick up both kinds of signals. SLS is static light scattering. It's the scattered light from those 2 lasers, and it gives information on when aggregates form. And DLS, its third technology, is the dynamic light scattering system and allows for detailed measurements of size and size distribution. These 3 detection methods can be operated isothermally at room temperature or across a heat ramp designed to thermally stress samples and see which ones take the heat. Uncle is able to deliver results on aggregation and 2 different looks at viral capsid stability in about 2 hours for up to 48 samples at a time. The Uncle sample holder is called the uni. Each uni has 16 wells, which are 9 microliters in volume. You'd load your sample into one of these 16 wells, just simply pipette. And anywhere from 1 to 16 can be used on a unit at a time. And then you seal it in the blue frame shown at the bottom of the slide between 2 silicone gaskets. The uni is what makes the Uncle truly unique, providing throughput and small sample size that works well for AAV samples. So Uncle can deliver 2 different kinds of stability information when it comes to AAV, capsid stability and aggregation. When it comes to capsid stability on the left of the slide, Uncle will use its fluorescence to measure 2 different behaviors. On top, you can see protein unfolding and capsid disruption occurring as a result, or on bottom, genome ejection can occur from mostly intact capsids. We'll look at data both from the literature and from Uncle to demonstrate these behaviors and investigate them further. With Uncle's SLS and DLS capabilities, we'll also be able to identify when aggregation occurs either at time 0 before an experiment to check that your sample is still nicely monodispersed, or as aggregation occurs during a thermal ramp as a measure of colloidal stability of your sample. Let's first dive into this model for AAV capsid stability and how we know that there are 2 possible ways for your capsid to degrade. In this paper by Verneau, the authors use atomic force microscopy, or AFM, to measure the 2 different stability pathways. To test this, the authors heated up AAVs serotype 8 or 9 to various temperatures and measured their morphologies. Shown at the top of the slide in the purple box, we see intact AAV capsids as bright white circles. As the samples were heated, the authors began to observe linearly ejected DNA coming out of intact capsids. Those are those wiggly lines coming out of the bright white circles in the green box. This is the genome ejection pathway that I mentioned. The authors also observed a third morphology where compact DNA was left behind in a sort of bundle or pile. The authors hypothesized that this corresponded to the capsid disruption pathway, where proteins would unfold and leave behind compact DNA. So how can Uncle figure out stability looking at these 2 pathways? Well, for capsid disruption, Uncle will look at the intrinsic fluorescence of AAV capsid proteins as these proteins unfold. Here we have a quick look at the raw intrinsic fluorescence signal collected by Uncle on a sample of AAV9. Each line is a fluorescence read at a different temperature. Uncle will excite fluorescence with its 266 nanometer UV laser, where we'll also collect SLS data at that same wavelength. When that laser excites the AAV capsid proteins, they will fluoresce in this intrinsic fluorescence range, between about 300 and 400 nanometers. As the temperature increases, the signal intensity drops and shifts to the right. So the blue arrow is meant to guide the eye to that shift, and that's helped by the dashed blue line showing the true vertical. Also note that you can see the SLS signal from the 266 nanometer laser. Over a heat ramp, Uncle will excite and read the full spectrum fluorescence of a sample at numerous temperatures. Here we're looking at the analysis of the previous fluorescence data. Along the y-axis on the left, we're tracking the intrinsic fluorescence as measured by the spectral center of mass, which we'll call the BCM, or the barycentric mean wavelength. You'll notice as temperature increases, the signal of that center of mass also increases, indicating that the fluorescence is shifting to the right. Where there's an inflection point in the curve, we've indicated a TM at about 78 degrees Celsius, and where the curve starts to increase, we've indicated a T onset temperature at about 75 degrees Celsius. If we take a look at the SLS data in green, we can look at when aggregation starts. So in this case, we're looking at SLS data for the 266 nanometer UV laser, which will increase when the particles start to aggregate and scatter more light. You'll notice that the onset for the signal of the SLS for the UV laser lines up very well with the onset for the unfolding behavior by intrinsic fluorescence. So with this one experiment, we're linking together unfolding of the capsid proteins with aggregation. Now let's take a look at genome ejection behavior, and that behavior is going to be at dramatically different temperatures. To study that other pathway for AAV, the genome ejection pathway, we're going to add cyber gold to our samples so that we'll know when DNA ejection is occurring. In this case, cyber gold has very low fluorescence in the absence of DNA, but increases in fluorescence as DNA is released. To track this behavior using UNCLE, we'll again use its full spectrum fluorescence. UNCLE will excite cyber gold using its 473 nanometer blue laser and will calculate the area under the curve for fluorescence. UNCLE will again use SLS to track aggregation of samples. Here's a look at the analysis for the cyber gold fluorescence experiment. In green, we'll be looking at the fluorescence of the cyber gold dye, and we'll be tracking the area under the curve. In blue, again, we have the SLS of the 473 nanometer blue laser. Okay, so starting at 25 degrees Celsius, we see some initial fluorescence signal coming from cyber gold because we know that we have actually free floating DNA present in this sample. As temperature increases, cyber gold fluorescence decreases, and we know that there's a relationship between temperature and cyber gold fluorescence, something easily shown with the DNA cyber gold control. However, the headline of this experiment is starting at about 50 degrees Celsius when we begin to see cyber gold fluorescence signal increasing, indicating that genome ejection is beginning to occur from our AAV capsid. We've also identified an inflection point at 58 degrees Celsius, which is what we'll use to compare different genome ejection curves until approximately 75 degrees, the inflection point will be used to compare different genome ejection curves across different serotypes and different formulation buffers anytime we run this experiment. And that behavior continues until about 75 degrees Celsius, where we again see our SLS signal spike, indicating that aggregation is occurring in our sample. Because we've also looked at the intrinsic fluorescence data, we know that aggregation, unfolding, and capsid disruption are all tied together for this AAV sample. In this one experiment, we're gathering info on how much initial free DNA is present, plotting the temperature where DNA release occurs, and the temperature when aggregation occurs, all valuable insight on an AAV sample. What we've seen from lots of thermal stability data on UNCLE and in the literature is that AAV has two different stability pathways that actually happen at two dramatically different temperatures. Capsid stability can be identified by intrinsic fluorescence and aggregation metrics and tends to happen above 70 degrees Celsius. Genome ejection is an independent pathway that happens as DNA is ejected from relatively intact capsids and can be monitored using DNA-sensitive dyes, like cybergold. It's important to measure both of these pathways to fully understand the behavior of your AAV. Here's an example of exactly that. So because buffers can also impact capsid disruption and genome ejection behavior, here we wanted to look at the results from an experiment on UNCLE, examining how stability changes in response to pH in an acetate buffer. You'll notice stability is highest at low pH, and then both behaviors show a decrease towards neutral pHs. Using UNCLE, this data was actually gathered in just 2 experimental runs, which is less than a day's total worth of work. Because buffers impact both behaviors unevenly, like this experiment shows, when optimizing formulation, it's important to measure both to know what's happening. So now with this UNCLE data, you can create an AAV that's ready for battle and won't aggregate or fall apart when under stress. Time to focus on Big Tuna, which simplifies and automates sample prep. Big Tuna is a fully automated, versatile buffer exchange system to help you power through buffer exchange, sample concentration, or sample cleanup. It can handle up to 96 samples in a single run with volumes as low as 100 microliters and all the way up to 48 milliliters. It's also designed to be super simple to use. All you need is about 30 minutes of prep time to design the experiment and prep everything on the deck. And Big Tuna will then take care of all your exchanges automatically, so you simply walk away and come back to finish samples. Big Tuna uses unfiltered plates as the key part of its sample prep. Unfilters have a regenerated cellulose molecular weight cutoff filter at their bottom, so that enables filtration in a plate-based format. Unfilters are available in different molecular weight cutoff options, so they're actually perfect for work with AAVs or any sample type. And they're available in different formats, unfiltered 96s and unfiltered 24s, so you can work with a wide range of sample numbers and volumes. Big Tuna works like this. For every cycle, the volume in each unfilter well is measured through a non-contact ultrasonic sensor, and then positive pressure forces sample through the molecular weight cutoff filter, while orbital mixing of the plate means the sample doesn't crowd at the membrane. Sample volume is then measured again, and then Big Tuna will add new buffer into each well. You just repeat this until you've reached your desired percent exchange, and you're done. And we've also recently launched new capabilities for Big Tuna to help make it even more useful for AAVs. So that means it can tackle the chore of buffer exchange from anything from AAVs to LNPs to mRNA to just more traditional therapeutic proteins. So for now, let's turn the dial over to AAV. When it comes to AAV, there's never enough. When it's produced, it's very dilute, so it needs to be concentrated to be used for characterization or just to get it to its final dosage form. Big Tuna can concentrate samples up to 100x. So let's take a look at how it does that. But very simply, to summarize how this experiment works, Big Tuna takes a sample and forces it through the molecular weight cutoff filter to concentrate it. Repeat this cycle over and over with more and more sample, and you can concentrate a sample multiple orders of magnitude. The workflow starts with 48 milliliters of sample that can be split across wells, say 1, 2, 3, and 4 in your sample reservoirs. And your goal is to concentrate those samples down to about 0.5 milliliter of final sample. So Big Tuna will concentrate, say, 1 sample in an unfiltered well, shown as number 5, since it continues to push buffer through the bottom of an unfilter and refill it from those 4 reservoir chambers. So numbers 1, 2, 3, and 4 will go into sample 5, and you can repeat that process for all 24 wells of an unfiltered 24. Our case study for this will be to concentrate AAV by 36-fold, and we'll use Stunner to characterize this experiment and give us a readout on titer and empty flow ratio. And here are the results. We're able to see that the initial AAV concentration of 4.8 times 10 to the 11th capsids per mil is concentrated beyond our target concentration to 1.9 times 10 to the 13th capsid per mil, with a final volume of 524 microliters. Our empty flow ratio is held quite steady, only increasing a bit, so we know our capsid payload is intact. We've achieved our 36-fold concentration, and importantly, we've done this in only 1.5 hours of walk-away time. So now, actually, we're going to pivot and focus in on our RNA LNP teammates, which is Stunner and Big Tuna. First, let's look at how Stunner delivers DLS data for LNPs while pinpointing total RNA quantification at the same time. One of the first things people notice about LNP samples is that they're really turbid, often so cloudy you can't see through them. Just like fog rolling in on the Golden Gate Bridge, you know that if only cloudiness were less of a problem, you could see some really spectacular things like, San Francisco. For LNPs, that would mean seeing size data without massive dilutions or getting RNA quantification by fast and convenient UV vis. Today, we'll look at how you can cut through that cloudiness in your RNA LNPs to gather some really spectacular data. With Stunner's RNA LNP app, Stunner can deliver low volume, high throughput DLS data without the need for lots of dilutions to get the size and PDI data that you need. On the same 2 microliters of sample, Stunner quantifies total RNA concentration without the need for any reagents, standards, or fluorescent dyes. All this is done in a 96-well, plate-based format that powers high-throughput analysis of LNP samples fresh out of mixing. We all know LNP size is critical to the performance of a successful drug. Throw a stick in the literature and you'll hit a paper with LNP size data. often showing the link between size and effective dose needed. So when you've got lots of buffers, formulations, and RNA constructs to test, you want to be gathering size data on everything. Stunner's DLS gives you the high throughput power to gather that size and size distribution data on up to 96 samples in less than an hour. Here we're looking at intensity distribution data for a whole plate of LNPs, with Z-average diameter noted as the value below each distribution. Across this whole plate, there's an average size of 79.8 nanometers with a 1.5% CV for these LNPs made using Genvoy ILM and generated with the generous help of Precision Nanosystems. With Stunner, you no longer have to be limited by DLS data that has to be manually gathered for about a dozen pieces of data in a day. Now DLS can keep up with LNP samples as fast as you can make them. All right, time to switch gears to UV-Vis. Anytime you try to see through solutions this cloudy, absorbance measurements done with plain vanilla instrumentation will have some issues to overcome. But load up this LNP sample into a Stunner sample well, and you'll quickly see it being wicked into the serpentine reservoir and held there. That's shown by the circuit on the left. To read the plate, Stunner pumps that sample into those 0.1 and 0.7 millimeter path length microcuvettes, and that enables UV-Vis absorbance measurements. When reading LNPs, the combination of Stunner's two fixed path lengths provides precision and broad dynamic range. And since LNPs are typically very turbid, cloudy samples, the short path lengths are needed so that light can pass through and enable absorbance measurements. Okay, so here's a quantitative look at all that turbidity, shown by comparing the signal from an empty LNP, one not loaded with a nucleic acid, to a PVS buffer. These LNPs create a whole lot of signal that looks like absorbance, but usually isn't helpful and just gets in the way. Because all that turbidity, still shown on the left, makes quantifying with traditional UV-Vis almost impossible, total RNA quant by UV-Vis is normally a big challenge. So if we squint at the full curve, we can actually see the RNA bump for these full LNPs at about 260 nanometers. But the difference between the curve on the right and the curve on the left is pretty small. You're not going to want to try to blank across different samples. So to analyze the curve on the right, we have to separate out the signal from the turbidity from the signal of the LNP absorbance due to RNA and other LNP components. On the left, we have that raw data from a full LNP again, but on the right, Stunner has used our Unmix algorithms to isolate the impact of turbidity and remove it. This is shown by the black line indicating total signal and the gray line indicating the contribution from turbidity. The remainder is in green and blue. Green is the isolated signal from RNA, and that will be our total RNA concentration. Blue is the UV-Vis signal from other components in the sample, UV-absorbing lipids, excipients, and buffer components. Stunner separates the contributions of those three groups, turbidity, RNA, and other components, to quantify total RNA concentration, all done in less than a minute per sample from only 2 microliters and without any dyes, reagents, or complicated workflows. Stunner's RNA concentrations agree closely with total mRNA concentration as determined by ribograde. On the left, we have a single data point, and on the right, we have a twofold dilution series of firefly luciferase mRNA LNPs showing highly linear results down to 1.2 microgram per mil. And Stunner still gets you everything all at once. Not only do you get DLS results on 96 samples of LNPs in about 1 hour, but from the same 2 microliters and in the same read, you get both size and your RNA concentration. So that's how Stunner delivers label-free, standard-free, and hassle-free gene therapy quant to the world of RNA LNPs. And with Stunner, you can cut through that fog finally and see all the spectacular details of your LNPs and maybe San Francisco too. All right, time to pivot again and look at Big Tuna, but this time how it can help with LNPs. In the world of lipid nanoparticles, a primary challenge is to remove the organic solvent that's left after the production of those LNPs. You want it removed as fast and as gently as possible to keep LNPs exactly how you made them. So that workflow might look something like this. We have an aqueous phase that contains the nucleic acid payload in green, and the organic phase that contains the lipids in blue. During production, Those are mixed, and we have an emulsion that contains both aqueous and organic solvents mixed in there. It's critical to take that through some kind of buffer exchange to clean things up, so it's just the buffer we want with our LNPs. And at the same time, it would be also ideal to control the concentration of the sample as needed. So this is where Big Tuna comes in. In this case study, we'll take a quick look at how Big Tuna exchanges LNPs starting in 10% ethanol into PBS. Big Tuna will also concentrate LNPs threefold at the same time, and all this is going to be done with 100 kilodalton molecular weight cutoff unfiltered 24. Big Tuna is able to handle all this work in about 90 minutes, and you can see the results here. Our sample concentration was successful, taking the RNA concentration from 26.1 microgram per mil to 83.5 microgram per mil, while maintaining good encapsulation efficiency, meaning that RNA was not lost to sample lysis or aggregation. Our target volume was hit, and our percent exchange value was hit as well, so we know we've gotten rid of more than 99% of our previous ethanol-containing buffer. All right, so that's a look at Unchained Labs' Gene Therapy Squad and how they can help out for all kinds of gene therapy challenges. So now let's pause and take some questions. But if you want to learn more about anything you saw here today, you can always reach out to us at info@unchainedlabs.com or come visit us at our booth in the exhibitor hall.
Thank you, Kevin, for taking us through the advantages of these instruments and how they can work together to address some key challenges in AAV and LNP characterization. We have a lot of great questions that have already come in, and it's not too late to submit them. Please type them, and we will get to as many of them as we can. So Kevin, I'm going to start with a couple of questions that came in on Stunner. First is, can you run one sample at a time on Stunner, and how long does it take?
Yeah, you absolutely can run one sample. You can run up to 96, but just one is fine as well. One sample takes about 45 seconds.
Okay, thanks, Kevin. And then does this work with SC-AAV, self-complementary AAVs?
Yes, yeah. So you'll notice that sometimes we talk about single-stranded DNA in our quantification, but it turns out when self-complementary AAV are packaged, then the whole Stunner algorithm works just as well. And I would even point back to that Stride Bio PCR slide comparing the ddPCR titers to show that, yeah, the quantification on Stunner still works regardless of if you're working with single-stranded or self-complementary.
Okay, thanks, Kevin. And another question on Stunner is, do you need to use known standards?
Good question. We get this one a lot. The answer to this is actually no. So Stunner is working from kind of a first principles approach based on all the information that it gathers, the DLS, SLS, and UV-Vis information. It takes really a whole look at all of that. It knows, based on the information you tell Stunner about this amino acid sequence and the DNA payload, it knows how much light scattering and how many it should expect and how many genomes you can make from UV-Vis. So it's really surprising the first time people run this that they don't need standards and they get results beyond their expectations.
Okay, thanks, Kevin. And there's one more in Stunner, and there's actually like 4 variants of this question, so I'm going to ask just one of them, which is, does the Stunner only work with purified samples, or would it also work with upstream sample types such as clarified lysates or supernatants?
So Stunner works best when you start to have purified samples, usually in kind of the column chromatography step of the process, possibly just after some depth filtration as well. And there's sort of 2 factors that are important there. One is the purity, but also the other one is the concentration limit, which we want to keep the concentration of the sample above about 10 to the 12th VG per mil, assuming you have a 4.7 kilobase genome. So when you're talking about concentrations like that, usually you're at downstream processing steps anyways.
Okay. And maybe let's do a couple uncle questions. One is for the experiments on uncle. Do different serotypes have different stability behavior?
Yes, absolutely. So that's been one of the very interesting things. And I would absolutely refer the audience, if you're interested in serotypes, to go check out our other app notes or webinars, because we take a look at a lot of them. We've run possibly every naturally occurring serotype now, and they all show dramatically different behavior, sometimes with 2 TMs, sometimes 3 TMs, often at different temperatures based on buffers. It's been really interesting kind of exploring the different serotypes and how dramatically they vary based on genome ejection.
Okay. And again on an uncle question, which is, there was a slide you showed which showed the capsid disruption starting to happen. And the question was, why is there not another increase in fluorescence when the capsid disruption started happening? And wouldn't you expect that to happen when DNA is released at that point?
So are you, I'm guessing that that question might refer to when we're looking at cyber gold fluorescence, would we expect a change around 75 degrees Celsius when we see protein unfolding as well? Is my understanding that correct?
That's my understanding of the question as well, yes.
Okay. So sometimes we do, and that's really just more of a result is because I believe the genome ejection has a kinetic component to it as well. So when you're kind of releasing DNA through your ramp, if you're ramping too fast or something like that, and your DNA frankly hasn't had time to catch up and equilibrate with the temperature that it's at, you could see behavior like that. But more typically, it's almost, you know, kind of linear if you're going at a slow, appropriate ramp rate.
Okay, thank you. And a couple Big Tuna questions. One is, what's the largest volume that you can concentrate on Big Tuna?
So the largest volume that you can concentrate on Big Tuna is going to be up to 48 milliliters of AAV if you're using one of the unfiltered 24-well plates. Okay.
And then another Big Tuna one, which is when you're concentrating samples on Big Tuna, how do you account for concentration stratification of the membrane? And then does this present a stability issue? If you want to
handle that,
I can handle this one, yeah.
You're more of the Big Tuna expert.
Yeah, I guess. So for Big Tuna, when we are doing a gentle orbital mixing when we're doing the exchange per step. So as a result, that means sample is not concentrating at the membrane. So the flow through the well is closer to what you'd have with TFF, which is that the sample is running perpendicular to the membrane surface. And as a result, there's two things. One is that the sample runs faster. And then two is we don't see a concentration gradient as we're doing the exchange, and therefore less stability issues of, say, aggregation occurring at the membrane. Okay. And one more Big Tuna question, which is what's the maximum number of samples and buffers that Big Tuna can buffer exchange in? I know you got this one.
So yeah, unfiltered 96s. So 96 samples, that can be, you know, if you want to pick 96 buffers, you're welcome to do that. If you want to pick 96 samples, you're welcome to do that instead. Okay.
I'm going to go back to some stunner questions because there's a lot of those, Kevin, and we have time. One is, does the stunner fill all requirements for assay qualification, and basically that's specificity, precision, linearity, et cetera, things like that?
So precision, yes, absolutely. Precision is something that Stunner is going to excel over most other comparable techniques. There's other presentations and data we have that can look at precision, especially for capsid titer, where you'll be pretty impressed relative to DLS and other ELISA techniques. For linearity, yep, as we've seen, it's highly linear. For specificity, Stunner is actually going to be, it's a biophysical technique based on size and then UV vis. So it's going to be kind of non-specific to capsid serotype, but everything else will be pretty much exactly what you need. And in terms of other requirements like validation standards, we have lots of standards available on the UV-Vis side of things. So you can look at accuracy, precision, linearity with our fundamental standards. We have IQ, OQ, PV services as well. So it's all kind of, once you start thinking about validation and IQ, OQ, PQ, we're ready for that too with Stunner. Okay.
And along the same lines of this type of questions, what is do we have an LOQ for titer determination for Stunner? What's a limit lowest detectable amount?
Yeah, so it'll depend a little bit on the serotype, the genome, the purity that you're using. But usually what we'll say is it's about 10 to the 12th VG per mil. That's assuming you have about a 4.7 kilobase genome.
Okay, and then maybe two questions kind of related to how Stunner is probably answered by how Stunner is doing the measurement, which is a question about if you're able to identify partial capsids with the Stunner.
So So basically, when Stunner's doing the analysis, it's going to divide everything up into two buckets, full and empty. So a partial capsid will have kind of a contribution to each of those buckets sort of proportionally. Yeah, so it can't distinguish partials on its own, but it will contribute those to the other sort of full and empty buckets.
Okay, and I think this probably is the same answer, but I'll just ask to make sure, which is for Stunner, can you detect over packaged capsids? So
yeah, same kind of answer. Yeah, it would sort of look as a heavier capsid, so slightly more than a full capsid.
Okay, and I think one more question, again, a Stunner one, which is, can Stunner be used to support release testing in a QC environment? Do we have examples of that yet?
We have customers that are working on that, but I can't say much more than that at this point.
Okay, I think that's all the time we have for questions today. So thank you again for submitting those questions, everyone. And Kevin, thanks again for a great presentation. So thank you, everyone who joined us live today. And if you would like to have a deeper understanding about any of the equipment we talked about today, please get in touch with us, as Kevin said, at info@unchainedlabs.com. We also have a virtual booth here at ASGCT, so you can check us out there. We're staffing that. We'll be happy to answer your questions there. We'd love to connect with you there or at any point down in the future. Thank you again for attending the presentation, and I hope you have a great rest of your day.